final one, that is conduction. So if I think in that way, if I just draw, then here this is like an ideal neuron. Okay, I'm not a good I have not a good hand in drawing, so like that. So this is your neuron or nerve cell. Okay, so this is the dendrite and this is the soma, and this is the hillock, and this is your Schwann cell, and this one is node of Ranvier. Okay, and this is the nerve terminal. So this is the ideal neuron. Basically, the action potential will travel very quickly through this type of nerve cell, because just here I want to mention the nerve cell has basically two types. One is myelinated nerve. Already I said this is the Schwann cell, and this is the myelin sheath. Okay, this is the axon. This is axon. So this is the myelin sheath. Myelin sheath means this is made up of a lipid in nature, and some proteins are there, but basically lipid. So it will act as an insulator. Okay, when action potential will travel, it will not be influenced by any external effects. Okay, so how and where action potential will create? So here the dendrite, through this dendrite your nerve cell will receive the information, and within the soma has some nasal bodies and some other organelles, and that will process the information, and that will just push backward to the hillock, and then it will travel to the terminal. So which form the information will travel? That is, electrical flow, and another one, the nerve terminal had some synaptic vesicle, and it will carry a particular neurotransmitter. Okay, so neurotransmitter, the chemical messenger. So to just cause the exocytosis, to cause the release of neurotransmitter, action potential, that is electricity, is a must. So where electricity will create? So I will just remember, already I said sodium channel, the touch sensitive sodium channel, voltage gated sodium channel, leaky sodium potassium channel. Leaky potassium channel is very rich. Then I said sodium potassium ATPase or sodium potassium pump. I said voltage gated sodium, voltage gated potassium. I said ligand gated ion channels. Okay, so where there? Basically, all the channels are there in dendrite and in soma, all types. But here, the voltage gated channels are very less amount. The soma and dendrite, voltage gated channels are there, but very less extent. Okay, so all types of channels are there, and over there, dendrite and soma, ligand gated ion channels are very prominent. Ligand gated ion channels are very prominent, and voltage gated channels are very less. Where? The soma and dendrite. Okay, and hillock, this is the trigger point of the nerve cell. Very sensitive, and there voltage gated sodium channel is very prominent, and voltage gated potassium also. So all other channels are there. The sodium potassium ATPase, that is the which is responsible for maintaining your membrane potential, is also present. Other ion channels are there, but over there in the hillock, the trigger zone of the nerve cell is highly prominent in sodium, voltage gated sodium channel. Okay, and there the node of NBR, that means you can say the gap between the Schwann cell. Within the gap, very prominent in voltage gated sodium and voltage gated potassium. The leak channels and sodium potassium pump are also there, but ligand gated ion channels are not prominent there. Okay, already I said ligand gated ion channels are very prominent in dendrite and stroma and also in the nerve terminal. Okay, but here the node of NBR are basically rich in voltage gated sodium and also voltage gated potassium, and some pump are there, but sodium voltage gated is very prominent. Okay, so what will happen if you just touch the nerve, or it will just give the stimulus of the nerve? It can at a time you can stimulate the nerve cell in a higher extent that will can create the threshold potential within the hillock, and it will create the action potential. Or you can touch here, and that will accumulate and it will reach the threshold. That means the accommodation, some summation of single or from that side, some extent of stimulus will aggregate and it will create the threshold level. That is possible. Or also you can like the punch in a strong way, and that will activate the nerve cell. Okay, so what will happen? So over there action potential can be possible. In general, action potential is not generating there, because exactly the action potential initiate the hillock, because their voltage gated sodium channel is very rich. But over there, in some extent that can happen, but usually not happen. So here, so all the some sub threshold potential or graded potential will accumulate, and that will activate the hillock point. Then what will happen? Over the hillock point, the voltage gated sodium channel will open, and then what will happen? Sodium will enter. You know, in the resting membrane potential, outside sodium is very rich, sodium is very higher than inside, so sodium will enter. And then when sodium will enter, then what will happen? It will create depolarization. So already I discussed depolarization. So depolarization happen by the entry of sodium through the voltage gated sodium channel, and instantly it will travel, because when sodium will enter, the intracellular sodium will travel through this hillock point to the next node of NBR. Sodium will just travel, and it will create the stimulus here, and that will trigger, or that will activate that node of NBR region, because in the node of NBR already I said voltage gated sodium channel and voltage gated potassium channel there. So what will happen? In which extent the potential change will happen through this inside, when depolarization happen in the hillock, the excessive amount of sodium will enter, and that will cause depolarization, and internal sodium that will travel to the next node of NBR, and it will activate or open the voltage gated sodium channel. And here the node of NBR, the voltage gated through the voltage gated sodium, again sodium will enter. Okay, so what will happen? Again sodium will enter, then here again depolarization will create. That means exactly action potential will generate in the hillock, and here action potential will regenerate. Okay, again here the internal or intracellular sodium will travel to the next node of NBR, and here the sodium will again enter. Okay, so here also again another potential changes, action potential. Here's another action potential. Okay, so action potential will travel very quickly as like jumping. Okay, because internal sodium will travel and some membrane potential will change, and that will combinedly will just activate the voltage gated sodium channel at the node of Ranvier, and that will create action potential, that is regeneration of action potential. So from this part to that part, again that part to that, it will travel towards the terminal. Okay, as like jumping. So this is called saltatory propagation of action potential, or you can say saltatory conduction. And this is happening very quickly because here no need to just maintain the resting potential. Just when sodium will just reach to the node of Ranvier, and that will open the voltage gated sodium, and sodium will rush into the nerve cell, and that will create the depolarization. And already I said where there depolarization will happen, instantly repolarization will happen. That means it will again try to reach to the resting membrane potential. This is the nature, okay, of the membrane, that will always maintain to resting membrane potential with the help of sodium potassium pump. Okay, that will happen, but actually through the depolarization, action potential will travel. Depolarization, because the highly activation part of the nerve cell, that is depolarization. If you want to activate the nerve, you must depolarize the nerve. Okay, so actually truly the depolarization is the main part for the activation of the nerve, that is action potential. Okay, so when the depolarization happen, instantly repolarization happen. Okay, so where depolarization already I mentioned several times, depolarization and depolarization, but after depolarization over there repolarization also happen. And how repolarization happen, already I said repolarization will happen by the exit of potassium, that is the repolarization. Okay, and it will maintain the resting membrane potential again through the sodium potassium pump and leak channel also. Okay, so basically there when it will travel, no hyperpolarization in a high extent, very little extent hyperpolarization happen. Very quickly depolarization, repolarization. Depolarization will travel towards the terminal, and repolarization happen after that. Okay, so over through this way, the nerve, through this nerve, through this axon, through the nerve fiber, very quickly electricity will travel towards the terminal. And then what will happen when the nerve terminal, the action potential will reach to the nerve terminal. The nerve terminal will contain large number of sodium channel, voltage gated calcium channel, voltage gated calcium channel, like that. Okay, so when the electricity will reach to the terminal, it will, here is the calcium channel, and calcium will enter through the calcium channel, and it will bind the synaptic vesicular surface calcium binding protein, and it will cause exocytosis, and it will release the neurotransmitter. Okay, that is the chemical messenger, and that neurotransmitter will just bind to the next, that is postsynaptic terminal of the nerve or effector organ, as you know. So my part is action potential, so I am just coming back to the action potential. So this type of action potential is very common because that type of neurotransmitter, that type of neuron is very common because this is the myelinated nerve. Okay, but in our body, some unmyelinated nerve also. That means no myelin sheath. You can say this myelinated nerve as like electric wire. Within the wire, the copper wire there, and cover is like plastic cover. Okay, so some nerves are unmyelinated, that is no plastic cover, only copper wire. Okay, such type of neuron, you can say some pain conduction, pain sensitive nerve, you can say thermal sensitive nerve, you can say some sensory nerve, which are basically unmyelinated. And that unmyelinated nerve, if you think like that, no myelin sheath, no myelin sheath. Okay, just I'm drawing here. So this is the halo, and this is like that. Okay, and throughout the nerve, the sodium channel, voltage gated sodium channel, potassium channel, sodium channel, potassium channel. Is there any gap? No, very continuously the channels are there. Okay, so what will happen when the halo will reach the threshold potential to generate the action potential? It will travel, and here action potential will create, here action potential will create, each and every channel will create action potential. And to maintain the resting potential, some energy will be required. Okay, so to restore the action membrane potential as well as to travel from hillock to the terminal, it will take more time. Okay, so this conduction is very slower, that is continuous conduction. This is very slow and it will take energy, and the saltatory conduction is very quick, and the continuous conduction. Okay, this is very slow, and saltatory conduction, you can say this is jumping conduction, very quick conduction, or propagation of action potential is very quick. And unmyelinated nerve, the action potential is very slow, and it will require energy, that will consume energy to restore the membrane potential for each and every part. Okay, so already I discussed the action potential generation, that is depolarization and repolarization. So just if you can think as like as saltatory conduction, that is conduction of the nerve. The conduction means, I said, generation, that is depolarization, that means it will create by the entry of sodium. Repolarization means it will create by the exit of potassium. So this is the same way, but the as here no myelin sheath, no node of Ranvier, so it will continuously happening, and it will create action potential that is very slow, and it will require energy. So in saltatory reaction, no energy required, and it will happen very quickly. So this is the action potential. So I'll just sum up the action potential. The membrane potential is always polarized in state in each and every cell, but when it will be functioning, it will just functioning means membrane potential is, also has some function to activate the intracellular signaling pathway like that. But when it will just travel in some particular muscles like skeletal muscle, smooth muscle, your cardiac muscle and nerve cell, that will travel, and that will basically, the membrane resting membrane potential, threshold potential, peak level, that is equivalent sodium level, equivalent potassium level, hyperpolarized state, like that. So overall, after stimulus, the optimum stimulus, if the potential changes will reach to the threshold level, only in that case action potential will generate. And before that, if you just touch the nerve and it will just slightly open the touch sensitive sodium channel, it will create some potential changes, but it is meaningless because it will be under the threshold level. So it is called graded potential or sub-threshold potential. When it will activate the nerve, you will activate the nerve in a certain extent that will reach to the threshold level, then real action potential will create. That in that point, voltage gated sodium channel will open and sodium will enter. And through the sodium entry, the current will just go towards the positivity. Okay, so that will reach the plus 30 or 35, in maximum extent 55 in some highly stimulus state. And then at that point, peak point, sodium channel will close and potassium, voltage gated potassium channel will open. And through this potassium channel, potassium will exit and it will create a repolarization. And that repolarization will reach to the resting membrane potential. Again, this is called repolarization. Okay. After that, through the leaked potassium channel, some potassium will continuously exit and it will create the potential changes below the resting membrane potential, and that is called hyperpolarization. Okay, and after hyperpolarization, the nerve again will reach to the resting membrane potential, and that is done by the sodium potassium 80 phase pump. Okay, in that way, the whole action potential will generate. So truly action potential means depolarization and repolarization, and you can consider as third phase of action potential, that is hyperpolarization. And already I mentioned the importance of it. Okay, and in that way, depolarization and repolarization, depolarization and repolarization, the nerve through the nerve cell, action potential will travel in myelinated nerve. It will travel very quickly as a jumping way, and unmyelinated nerve, it will travel slowly because frequently action potential will generate and it will travel very slowly and it will require energy. Okay, so action potential is very important. If you consider, already I discussed regarding the nerve cell, so neurotransmitter release action potential is important. Without action potential, nerve cell terminal will never release neurotransmitter. That is electrochemical signal. Okay, so electrical and chemical. But if you think about cardiac muscle, no action potential, no heart muscle contraction. If you think about skeletal muscle, not like that. If you think about the blood vessel, contraction and relaxation is needed. So such type of function, if you want to get, action potential is important. So some extent in nerve cell and cardiac muscle, action potential mechanism somewhat different, but the basic thing is same. Okay, so this is the action potential, whole action potential in a row. So thank you very much.